Hi, I'm Mr. Baker, and this is 8.1 and 8.2, the intro to Mendelian genetics. To understand Mendelian genetics, you have to learn about Gregor Mendel, who's the father of genetics, or the study of heredity is how that word is pronounced. He was a monk back during the Civil War era, except he was in what is now the Czech Republic, and he studied how pea plants passed traits to their offspring. There were 7 traits in pea plants that he followed, and he crossbred those plants with the different traits, and he made conclusions from the results. He looked at plump versus wrinkled pods, and round versus wrinkled peas, and look at colors and flowers, and tall versus short. He came up with the idea of 3 generations, P standing for the first or parental, F1, which stands for first filial, and you can just call that the kids, and then the F2, the second filial generation, which is, you can just call them the grandkids. You need to understand the difference between traits and alleles. Mendel was working long before there was an understanding of what chromosomes were, what DNA was, what genes do, and how all this works. So he used the term trait. And trait is a characteristic of a living thing, or you can substitute in gene as far as I am concerned in my class. And of those genes, you can have different forms of that genes, and he called those alleles. And you might have 2 or more alleles per trait. For example, hair color. You can have hair color that's black, brown, blonde, red, and some other colors perhaps. But the one that I really want you to focus on is the eye color. Eye color, and there are multiple eye colors, but we're only going to look at blue and brown because eye color is the trait, and there are 2 alleles that we will play with when we set up our Punnett squares later. And you can either have a gene that tells you to be blue-eyed, or you can have a gene that tells you to be brown-eyed. So make sure you understand the difference between trait and allele. That is a karyotype, or a picture of the chromosomes, and this is a male because you can see down at the bottom he's got an X and a Y chromosome. The Y there is the green dude. Alleles come in pairs, and typically they're going to be or have one that is dominant and one that is recessive. The dominant one is the strong allele, and if you have that dominant allele for a given trait, it will appear no matter what. The recessive is the weak allele, and if you have a recessive allele, it can be hidden by the dominant allele. Any organism possesses 2 alleles for every trait because you get one from mom, one from dad. Just like we learned you get 1 chromosome from mom and 1 chromosome from dad. These are represented by dominant alleles being capitalized and the recessive allele being the lowercase letter. You need to know the terms homozygous and heterozygous. If you're homozygous, you have 2 of the same allele. For example, and I'm going to use big and little. Big A is homozygous dominant, meaning you have 2 of the dominant. Little a is homozygous recessive, meaning you have 2 little or 2 recessive alleles. Heterozygous is when you have the 2 different alleles, big A, little a. The dominant allele will win because it's the stronger of the 2. And you can also refer to a big A, little a as a hybrid, or heterozygous are also hybrids. You need to know what a genotype versus a phenotype is. A genotype are the allele pairs in the letter form, such as big A, little a, or little a, little a. The phenotype is what you can see. Again, they were making up these terms way back when, before they knew what genes, chromosomes, and DNA all did. So, for example, the genotype is big T, the phenotype is tall. The genotype, big T, little t, those are the letters. The phenotype is still tall because the dominant wins. And if you've got a little t genotype, you are short. That's the information for 8.1. 8.2 is where we are now, and that includes the law of segregation. There's a whole lot of big definitions for this, but the big thing is it really distills down that you get one of the traits from mom, one of the alleles from mom and one of the alleles from dad. So that translates to you get 1 chromatid from mom and 1 chromatid from dad. So if you see anything about the law of segregation, it's you get 1 allele from mom and 1 allele from dad. And there's a picture that represents that we covered in class. You need to know what a Punnett square is. It's a 4-square box. It's a technique used to predict the possible offsprings of a cross between two organisms. And each square represents one of the possible genotypes of the offspring. So for example, if I have a big R, little r, that mates a big R, little r, and you fill in the box. You'll be shown how to do this in class. A monohybrid cross involves only one trait, and that is a 4-square box. For example, height. Here we have 2 alleles. The big T equals tall, the little t equals short, and you fill in the box that way. We will film this in during class, and I'll show you how to do that. And here's another example that we'll do during class. And here's one that shows you what's going on with hairy and hairless guinea pigs, showing that you've got a big H, little h, crossed with a big H, little h, and what the results are. So watch that a few times. There is another law you'll need to understand. That's the law of independent assortment. And this says that different traits are inherent independently of one another. The gene controlling the color of your eyes has little or nothing to do with the gene controlling your height. Simply put, 2 different genes are most likely found on different chromosomes. That leads us to something else you'll need to know, the dihybrid cross. This involves 2 traits, and it's not a 4-square box, it's a 16-square box. Example, height and eye color. You have 2 alleles for height, and you have 2 alleles for eye color. We're going to do tons of practice problems. But you'll need to understand the FOIL method, which is from math, and it's first, outside, inside, and last. Your 16-square box will look like this. I have that color-coded for a reason that I'll explain in class. And that takes us to things you need to know for quiz 8.2. The notes will just be over the notes, and you will have separate quizzes on how to do Punnett squares. All right, study hard, and I will talk to you next time.